Okay, well, first of all, you're welcome for the sun. I just brought it, although it's trying to go away. I want to thank the foundation for inviting me to speak to you today. And we're going to talk about a little bit about our mouse model and then our organoid model. And then in the last part, we've started testing potential antisense oligonucleotide therapy. So our mouse model was developed by Julie Zebro, a pediatric epileptologist in the lab, in collaboration with Hisashi Umemori, who generously gave us his PCDH19 knockout mouse that he's published, had a science paper a few years ago. And then Sundeep Kalantri gave us an XGFP mouse model. So the idea is we cross the knockout with a female with a male XGFP mouse, and in the resulting heterozygous females, the wild type PCDH19 goes along with the GFP allele on the X chromosome. So the GFP labeled is wild type, and then the knockout has no GFP. So it lets us track it. And then the males are hemizygous with no GFP. And for the control, we cross the wild type with the XGFP mouse so that some of the, by random X inactivation, about half have GFP and half are unlabeled, but they're all wild type. So on the right, you see what the wild type looks like. This is at postnatal day 1, where GFP is everywhere. And then in the heterozygous female knockout, you see stripes of wild type cells and then unlabeled knockout cells, similar to what Paul Thomas first presented with his model. So that's in cortex. At P45, we're also seeing it in hippocampus, predominantly in CA1, where you have areas that are knockout mixed with areas that are wild type. We don't really see segregation or stripes in the dentate gyrus in CA3. And Hisashi saw the same thing in his mouse, mainly CA1. Not clear why that happens. So Julie looked at interneurons in both the cortex and hippocampus and didn't see any differences in the heterozygous female in the cortex, but in area CA1 of the hippocampus, she saw a decrease in parvalbumin interneurons that you can see here. Here's the wild type and the heterozygous female, but no difference in somatostatin or total GABA in CA1. And then Yukun Yuan and Lori Isom's lab did some slice physiology recordings, and these are preliminary, but at P16, some of the mice showed a hyperexcitability phenotype with these doublets that we've seen in other DEE mouse models that interestingly goes away at P31, which is why one of the reasons I was wondering if there's a developmental effect. And then Julie did hyperthermic seizures. We put the mice in this contraption and heat them up. And when we look at seizure severity by the Racine score, which higher means more severe seizures, we see a significant increase in heterozygous females compared to both wild type and knockout male model. We also see earlier onset of seizures at lower temperatures in the heterozygous female, which was only significant compared to the wild type, not compared to the knockout male. So this is done at P16. If we do it in young adult mice, we don't see a phenotype also, which is made me wonder if there's a developmental effect. So we looked early. I think Paul saw cell sorting as early as P9 or P10. Here, I mean E9 or E10 in the embryonic brain. Here we're looking at E12.5. And in addition to cell sorting in the cortex, we see it in the ganglionic eminences, which made us wonder if the decrease in parvalbumin interneurons is from an early developmental effect of interneuron development in the ganglionic eminences. And Julie's starting to look at changes to interneuron development. Here, she looked at proliferating cells in the ganglionic eminence, comparing GFP positive wild type cells with GFP negative knockout cells in the mosaic. And in wild type, there was no difference in Ki67, the marker of cell proliferation in the wild type versus the knockout cells, but in the heterozygous female, more of the wild type cells are proliferating than the knockout cells, suggesting this could be one of the reasons for the later decrease in interneurons. So I'm going to move on to the organoid model. So in making brain organoids, the typical protocol starts by aggregating human pluripotent stem cells, either induced pluripotent stem cells or embryonic stem cells, into embryoid bodies, and you go from this 3D structure into a 3D brain organoid structure, either via unguided differentiation, where you get lots of tissue, different types of tissue, but it's very inconsistent. So most people use a guided differentiation using small molecules to make brain region specific organoids. And then one can differentiate them into different regions and combine them with fusion into what's called assembloids. So for example, you can fuse cortex and thalamus and look at connections between the two or cell migration. And I'll talk a little bit about fusions later. But all of these techniques have these what we call multi-rosette organoids. And each of these neural rosette structures represents the neural tube. But as you know, normally in development, we start with a neural plate that's 2D that folds into a 3D neural tube structure. So to try to model this more faithfully with our brain organoid protocol, Andrew Tidball developed a new method. So he was a postdoc in the lab. He's now a research assistant professor. And he came up with the idea of using a monolayer instead of embryoid bodies. And other tissue organoids start with a monolayer. So he cuts them into uniform squares, placed them in Geltrex, and they round up to form these single rosette brain organoids that we call saucers, self-organizing single rosette spheroids. They have very consistent growth rates. You can see them growing here between day 9 and 18. And they generate the normal complement of progenitor cells. So they generate VZ-like cells that express nestin and PAX6, SVZ-like cells that express TBR2. Here we see young neurons at the outside that are double cortin positive by day 22. And then by 5 months, they're generating the full complement of 6 cortical layers. So by 8 weeks, we see outer radioglia that express HOPX that you can see nicely organized here. And then they express deep layer neurons that are CTIP2 positive, intermediate layer neurons that are SATB2 layer 4, and then CUX1 and BRN2 layer 2-3 neurons. They also make reelin. We've done single cell RNA-seq at 1, 3, and 5 months. The organoids are very consistent, and they show markers of all 6 cortical layers. So we asked, can we model PCDH19 clustering epilepsy using this single rosette model? And so Wei Nu, a research assistant professor in the lab, and Sandra Mojico Perez, a star technician, generated human embryonic stem cells with knockout of PCDH19. And they labeled them with RFP or GFP, and they also labeled wild type with RFP and GFP. And the idea is, if you mix wild type and wild type, or knockout and knockout, you shouldn't see a cell sorting phenotype. You should only see it in the mosaic situation. And with wild type mix, they mix nicely. Knockout and knockout mixes nicely, but it's only when we mix the wild type and knockout that we see the cell segregation. Wei quantified this, and again, with wild type only or knockout only, you don't see the striping, you only see it with the mosaic. We've done this with male cells to model a somatic mutation in a male. So these are iPS cells with CRISPR knockout of PCDH19, and the knockout is unlabeled. The isogenic control expresses GFP, and this is live imaging with a within incubator microscope. And you can see the cell sorting here compared to the control mix where you don't see it. So it's very consistent and robust. We also compared a multi-rosette protocol with our sausage protocol. So this is spin omega. Guoli Ming and Hongjun Song developed this organoid protocol. And these are bigger than our saucers because we only have a single rosette. They have multiple rosettes. You can see with zona occludens 1 is labeling the inside of each of the rosettes here. And you can't see the cell sorting phenotype very well in a multi-rosette protocol compared to the single rosette. Again, this is male with knockout unlabeled and wild type labeled. We have also mixed different ratios to try to model kind of different amounts of skewing of exon activation, and mix 1 and 2 are wild type, and knockout. And again, we don't see the striping. With mix 3, 4, and 5, which are 1-to-1 or 2-to-1 or 1-to-2, we see striping. We start losing it a little bit when we skew it 1-to-4 or 4-to-1, as you can see here. So it fits, it makes sense, and it'll be nice to look at later phenotypes to see if they decrease with the skewing. Early cortical development in the mosaic saucers is altered. So here we're looking at CTIP2, which is a marker of early layer neurons. Ignore the inside because that's kind of cortex coming into the picture, but this is really the rosette outside it. When we look at the layering of the early born neurons in wild type or all knockout, it's pretty organized, whereas in the heterozygous situation or the mosaic situation with wild type and knockout cells, the CTIP2 is disorganized either because of abnormal migration or premature differentiation. I don't have time to show you, but we also found alterations in cell proliferation, NCAD expression, and subcellular localization of NCAD and PCDH19. So we put these mosaic or knockout rosettes on multi-electrode organoids on multi-electrode arrays for recording and compared them to wild type. And when we look at mean firing rate or burst frequency, the wild type is much more active than the knockout only or the mosaic, similar to what Sarah showed in terms of hypoactivity. Now this is only the dorsal excitatory organoids. So we're starting to make ventral ganglionic eminence organoids to look at the phenotype of the interneuron progenitors. And most ventral organoid protocols that actually not most, all of them that people have generated, they call subpallial or ganglionic eminence because they're not just making medial ganglionic eminence or caudal ganglionic eminence, but they also make lateral ganglionic eminence, preoptic area, and hypothalamus. So it's kind of, the ventral ones are dirty. So we played around quite a bit to try to optimize it just to make MGE, mainly by changing the timing of the SAG treatment, smooth antagonist. So when we did it originally at day 6, we got a mix of all these different areas. But if we give it at day 10, the organoids don't like it as much. They're initially not quite as healthy, but then they develop nicely and they make markers just of MGE, including LHX6 and KX2.1. And they're not making RX or PAX6, which you can see in hypothalamus. So we think by QRT-PCR also that they take on an MGE-like phenotype, which is what we want because they make the parvalbumin and somatostatin interneurons. So we looked at cell sorting in the ventral, and this is again live imaging with the incucyte within incubator microscope. It's not quite as robust as the dorsal organoids, but we do see cell sorting compared to wild type, and knockout mixes shown here. either the knockout is GFP labeled or RFP labeled, and we can see the striping pattern. So we want to also look at interneuron development, including migration. And as many of you know, the MGE cells migrate tangentially into the cortex to integrate with excitatory neurons. Once they get there, they do some radial migration laterally. And so to model that, we make assembloids or fusions where we take MGE patterned organoids and excitatory cortex patterned organoids and fuse them. Here we fused at day 78 and looked at day 125, and it's a static image. But when you look at the CTIP2, which is an excitatory deep layer marker, it's only in the dorsal, not in the ventral. And when you look at GABA, it's in the ventral, and putatively these cells are migrating out into the dorsal organoid. So we're looking at different timing of fusion to try to optimize it. Here we fused at day 54 and imaged 5 days later. And let me see, shoot. Oh, there we go. So you can see the GFP labeled cells are in the ventral and they're migrating into the dorsal, just as expected. The interneuron progenitors migrating to the excitatory domains. And it works very well. Here we've taken either mosaic knockout and wild type PCDH19 ventral fused with a mosaic dorsal, or all knockout in the ventral and dorsal. But these were fused at day 26 and imaged 4 days later. And we see some migration, but it's not profound. So we're now repeating these studies at later time points to look to see if we see a differential phenotype with loss of protocadherin in the interneurons. All right, so just to summarize, our PCE mouse model shows structural and behavioral phenotypes, including cell sorting in both dorsal and ventral progenitor domains, decreased CA1 parvalbumin interneurons, and a lower hyperthermic seizure threshold and increased seizure severity. Our new brain organoid model successfully models cell sorting phenotypes in dorsal more than ventral, and we also see abnormal early cortical development in the dorsal organoids. And current work is exploring interneuron development in the fusion organoids, looking at synaptogenesis, and we're looking at network function in both models. While we see dorsal only organoids plated on the MEAs, we see decreased activity. We're hypothesizing that in our fusion organoids, which actually produce better activity because the interneurons help the excitatory neurons mature and the excitatory neurons help the interneurons mature, we're expecting we may see hyperexcitability in the fusion mosaic model. All right, in the last part of the talk, I'm going to show you our ASO work, which is also at a preliminary stage. But the idea is, can we prevent or reverse the PCE phenotypes by decreasing wild type PCDH19 to model a hemizygous male state? So all of you pretty much know this. One way that ASOs work is by forming a heteroduplex with mRNA and inducing RNase-H to break down the mRNA so you don't make protein. And we collaborated with Jackie O'Rourke and Barrett Powers at Ionis Pharmaceuticals. They generated lots of different ASOs targeting PCDH19, first in mouse, and they screened them. Here they injected ICV, the ASO at P1, and looked at 2-week-old mice and found a lot of ASOs that knocked down PCDH19. And they also looked at toxicity, looking at GFAP staining and markers of microglia. And we found 2 that did nice knockdown without inducing inflammation. And they sent these to us, and Julie injected them at P1 or P2 intracerebroventricularly in our PCE mouse model, and then harvested the tissue or did hyperthermia at P16. And by qRT-PCR, we see nice knockdown with both of these ASOs in both in our heterozygous mouse model. And in the hyperthermic seizure model, we see a hint of an effect. but we're only at 10 mice per group at this time point. But it may, compared to control ASO, it may reduce the seizure severity. The other thing I forgot to mention when I showed the hyperthermia model is those data were without EEG recordings, but we see some non-convulsive electrographic seizures when we do EEG recordings during hyperthermia. So we're repeating the analysis just to look at EEG documented seizures, and I think that'll maybe help us get a more robust, see a more robust effect. So it seems to work to knock down PCDH19 in mice. We're also applying the ASO to the brain organoid model. Ionis tested it in a different human neuronal cell line and also did an LDH assay for toxicity. So here we've generated our saucer's dorsal model and given either 40 micromolar ASO starting at day 23 and looking at day 38, or 20 micromolar starting at day 60 and looking at day 89. And we're giving it every other day with each media change. ASO1 seems to cause a lot of toxicity. You can see the cell death around the organoid. ASO2 is better tolerated. Here at day 38, after 16 days of treatment, ASO1 and 2 both show a nice knockdown by qPCR and by Western blot. It's not quite 90%, but it's a substantial knockdown. In the second experiment where we gave it day 60 to day 89, we see a much better knockdown here by PCR and Western blot. So we think we can knock it down pretty nicely. But our phenotype is in early organoids. Actually, before I get to that, this is an experiment. Hopefully this will play, maybe not. No, I can't do that. Oops. Oop. All right, I'm gonna go backwards and forwards. All right, let's see if this one will start playing. Here we gave a Psi3 labeled antisense oligonucleotide directed at mallet1. Here, they're both playing. Here, on the left is a single treatment, and you can see over 20 days, you start losing the ASO, but here we're giving it twice weekly, and we get nice persistent expression of the ASO. So we think it's getting in, we think it's doing what it's supposed to do if we give it at least twice weekly. And what Wei did was start treatment at day 6 of our organoid model, day 10 or day 12, with 20 micromolar of ASO twice a week. We got nice knockdown, but when we look at the growth, the 6 and 10 day, and less so the 12-day treatment, decreased the growth of the organoids, so it's having an effect. We don't know if it's on target, off target, or some other backbone toxicity, but you can see the ASOs are much smaller. We don't see here you can see the striping in the untreated that we don't see in these, but we don't know if it's because they're sick or if it's really working. Day 12, we actually start seeing some cell sorting, so that may be too late to prevent the phenotype. I should say we don't see the phenotype until about day 16 or 18 of organoid development. So we're currently exploring lower dose ASO and less frequent dosing in our early saucers, and then we know ASO2 was not toxic in the late organoids, so we're looking for later phenotypes that we can assay. But I think we're excited. I think this is going to be a nice approach combining both the mouse and the organoid model. So just to summarize, PCE mice display cell-sorting hyperthermic seizure and PV interneuron phenotypes. The brain organoids exhibit cell-sorting, deep layer neuronal development and hypoexcitability phenotypes, and the fusion model should provide insight into interneuron development, synaptogenesis, and function. And our ASOs targeting PCDH19 significantly decrease protein levels in neonatal mice and in the brain organoids, and I think should offer a potential therapy if we see it works in these preclinical studies. So with that, I tried to acknowledge the people as I went along who did the work, and I want to thank the foundation and Safari for funding ASO study. So thank you for your attention.
We have 10 minutes for some questions if you want. So.
I was wondering, so there are a couple of studies out there who've done single-cell RNA-seq on human samples of different developmental stages of the cortex, and interestingly, they don't seem to show expression of protein 19 in human genitals. So I was wondering, in your in vitro system, you do seem to have clear protein 19 expression there. So
yeah,
have
so
you looked at those studies, and do you know how could correlate?
yeah, so when we do 2D neural rosette to neuron differentiation of human pluripotent stem cells, PCDH19 goes way up in the neural rosettes, and then it goes down in the neurons, but it's still much higher than in the undifferentiated pluripotent stem cells. So we definitely see expression. Eventually it gets probably into specific layers, and so some of those other studies may not be looking at the right layers.
Beautiful, Jack. I'm still trying to all comprehend it, but for your hypertonic model, I think we tried this whole with Steve Becher, but we probably tried too late. So have you looked at later stages?
Yeah, we don't. You
didn't see anything.
We didn't see a phenotype at P40. I think we looked.
P40.
Yeah, and then when we did. I can't remember if Julie did PTZ or pilocarpine to look at seizure thresholds. When she did around P40, if anything, they were resistant and didn't have a decreased threshold. So
that makes sense. Now, when I look at the distribution of your individual mouse scores, there are certainly many mice which are exactly like wild type versus knockout. Given that you have those mice, given that you can visualize the axon activation and the PCDH19 plus minus status, did you look back at some of those mice which shows very high sensitivity to temperature to the mice who didn't show any difference with respect to the wild type, knockout situation, whether they have a different patterns of distribution of the axon activation of the cells?
Yeah, no, excellent.
Trying to correlate it doesn't make sense.
Excellent question. Julie tried to correlate just looking at hip stripes in hippocampal CA1, and could not correlate it, but that doesn't take into account.
Not in hippocampus, no.
It didn't correlate with the hippocampal cell sorting, but that doesn't take into account cortex, and so it's hard to know the right place to look, and it's kind of messy. So I think where we're heading is to look at slices and look at excitability of the GFP labeled versus unlabeled cells at boundaries of the stripes versus away from the boundary, and then try to do calcium imaging to look at the connectivity in slices. So hopefully that'll show something.
That's interesting, because perhaps it may depend on which part of the brain is controlling the temperature, if that's the temperature control, or it's some other response where probably the mosaicism might be more relevant.
Yeah, and there's nothing that really suggests that the cell sorting itself is related to the phenotype, and it may be totally unrelated and all synaptogenesis, a later synaptogenesis phenotype and connectivity. So we have a lot to learn still.
If I may have another question. I think some time ago we have done very limited experiments with the differentiation of the iPS cells derived from a male with PCDH19 mutations versus controls. And there were different size. The rosettes were different size and shape. The lumen was slightly smaller, which we consider to be just a difference between the knockout versus wild type. But did you look at did you compare your rosettes and the sizes and shapes and the lumen, whether there were any differences, even between wild type, knockout, if not the heads?
Yeah, so when we started this long, challenging process of studying this very difficult disease to study. A grad student who was in the lab, Cece, was starting with patient material and doing 2D rosettes and thought that the lumen sizes were different between patient and control. But in any patient culture, you never know the degree of exon activation, and it is sloppy. And so when we started mixing with the mosaics, we didn't see that. And we've worked with well over 100 different human pluripotent stem cell lines, and there are line-to-line differences in the rosettes. So it's hard to.
It's another rock.
Yeah. So we have not been able to recapitulate that.
But the premature differentiation kind of fits what we have seen.
Yeah.
We have seen in these mouse neuronal stem cells that seems to be kind of a.
Yeah, that's robust.
But that's also a kind of a developmental phenotype, which then not necessarily good.
Yeah.
Can I say something about that? Because we've done a very thorough characterization of one of the proto-19 local models. And the interesting bit there is that there are no changes in the number, or no major changes in the number of positioning of cells. So I think there's a chance that in vivo there might be other regulatory mechanisms that can maybe compensate for that you lose when you put the cells in vitro and you have a more artificial system. Because given the information out there, we would have expected to see more differences, maybe in position of cells, as you see, for example, in your organoids, and that doesn't seem to be the case in the mouse model. Right.
So, yeah, a couple of things. So, that early phenotype, we don't know how that translates later. Organoid models for many DEEs or other neurodevelopmental disorders show more severe phenotypes than mouse models. And much of that is probably the stress of an in vitro system, but then there are also potentially mouse-human differences that we don't know about. So, but that's a good point, yeah. All right, thank you.